is everybody today? I hope everyone is all right out there and having a good start to the week. I'm starting to just start with a little bit of updates and news and all that kind of stuff. And I'm sorry to kind of start with bad news, but I want everybody to kind of know what's going on with Ocean Stories and why we haven't released a schedule for the coming weeks yet. Well, starting next week, Ocean Stories is going to begin cutting down how many episodes we actually publish each week due to budget constraints. We're going to be announcing 2 new talks for next week later today, and then potentially moving to 1 talk per week or possibly 2 starting the first week of June. The latest news updates from the government here in Mexico aren't really looking that great for Baja at the moment, so this kind of means Dive Ninjas has to kind of cut some excess spending and all that kind of stuff so they can keep moving on. But we're going to keep working hard and see what we can do to try to keep Ocean Stories running for as long as possible and get as many episodes up as we can, physically do. However, this week we still have 2 more incredible talks coming up. On Thursday, we have Martin, who's one of the founders of Coral Guardian, joining us to talk about their work with coral reef conservation in Indonesia. And then on Friday, we have a super special talk with Jack Fishman. He's the Community Conservation Officer of Project Aware. He's going to join us to talk about a little bit about the remarkable work they're doing around the world right now with divers and communities and all that in conservation. But he's also going to talk a bit about how us ocean lovers can actually still get involved in conservation while we're all quarantined and stuck at home and all that kind of stuff. But first, today, we have one of my favorite humans joining us, Marta. Marta's a marine biologist from Spain. She's done an incredible array of work, from studying freshwater dolphins in the Amazon, to penguins in New Zealand, to monitoring coral reefs in Indonesia, assessing environmental impacts of divers in Galapagos, and the list just goes on and on. But she's also part of our family, of the Dive Ninja family, and is the lead scientist on our Mobula Ray citizen science expeditions here in Baja. So how are you today, Marta?
Hi, Jay. Good.
Awesome. We're super happy to have you here and everything like that. So let me stop sharing my screen and I will hand it over to you. I'm also going to start the poll real quick. I'll launch that. Cool. The poll is launched. There we go. You should be able to share your screen now.
Okay. There you go. Can you see me?
Perfect. We can see you. I'll let you take over.
Okay. So, hi everyone. I'm Marta D. Palacios. Today I'm going to talk a little bit about the mobulas of the Sea of Cortez. As Jay has explained you, I've been working with Mobula Rays since 2017. But before, I was researching oceanic mantas in the Galapagos Islands with Manta Trust and the Charles Darwin Foundation. And after that time, I realized that the Mobula Rays, the small portions of the manta rays, have very little attention from the researchers. So I decided to move to Baja California and start a project with the mobulas. And that is how I ended up doing my master's, focusing on one of the species of mobulas that we have here. But of course, it's not just me that it's going out to the field and tagging the animals and doing all the things. I have a super team of local fishermen from the island of El Pardito, Juan and Felipe, and students from the University of Baja California Sur, and then a lot of researchers from the University of California, CICIMAR, Palacios, and other institutions. So first of all, I will introduce you a little bit of what is a Mobula Ray. So Mobula Rays are elasmobranch. That means that they don't have bones as the rest of the fish. They have cartilage. So they are negatively buoyant. If they stop swimming, they will sink. They belong to the rays. So we have sharks and rays. So inside of the rays, we have the benthic rays that live all their lives close to the bottom. And then we have the pelagic rays that are the mobulas and the manta rays. These rays live all the time on the column of the water, like swimming, free swimming, and a very bad reputation that they have, and it's not true, is that they don't have a sting or if they have, they don't use it. So they are harmless, not as the stingrays that we have normally here at the beaches. So they are also filter feeders. That means that they feed on zooplankton that is floating on the water, and they are super smart. They have the biggest brain of all fishes, and they have areas of the brain that are highly developed that increase their sensory functions. But there's always like a misconception of manta and mobula. So mantas are the most famous ones. There are 3 species, and they can reach up to 7 meters of this width. That means from here, you can see my pointer, from one tip to the other tip of the pectoral fins. That can reach 7 meters. And then the mantas have their mouth on the front side of their heads, while the mobulas have their mouth on the ventral part. Also, here you can see the cephalic fins of the mantas. Those cephalic fins are used to funnel the water towards their mouth. So when they are feeding, they just open the mouth and roll the cephalic fins and start to feed on zooplankton. So mobulas have smaller cephalic fins, and when they roll them, they look like horns. That's why they are also known as devil rays. For mobulas, we have 8 species, and the biggest one is the one here in the picture, is the tarapacana, and it can reach up to 3.5 meters of this weight. So here in the Sea of Cortez, I like to call it like the epicenter of the mobula world, because we have 5 species. We have 1 species of manta, that is Manta D, and 4 other species of mobulas. As you can see, they are all listed in the red list of the UCN as endangered or vulnerable. So 4 of these species, the Mobula thurstonii, japanica, or Mobula tarapacana, and birostris, are found in every tropical, subtropical, and warm temperate oceans of the world. Just the exception of our superstar, the Mobula munkiana. They are only found in the eastern tropical Pacific. So this ray is very famous because they form huge aggregations of tens of thousands of animals swimming at the same time, and it's very spectacular to see. And also what they do is they like to jump a lot. So jumping for Mobula rays can be some sort of communication. So that's why these animals, the Mobula munkiana, is like the most social mobula, and that's why they are jumping all the time. So Mobula munkiana, they are born with 35 centimeters of this white, so very, very small. And after like long time, we still don't know for how long they can live, but they can reach up to 1 meter and 20 centimeters or even 1.5 meters. So why all these mobulas and mantas are endangered or vulnerable? It's because they have a very conservative history trait. That means that they reproduce very slow. They have pregnancies of 12 months, and for every pregnancy, they just have 1 pup. Their reproductive cycles are every 2 or 5 years, they have a baby. So imagine for the entire life of a mobula, they will have, if they are doing well, like 14 babies, and that is the number that a great white has just on 1 liter. So that's why they are very, very vulnerable to any kind of overexploitation. Also, they reach sexual maturity very late, and the studies on their longevity, they say between 14 to 31 years, but it's not like true. So all these things make that in many countries, as in Mexico, they are protected, and it's illegal to fish them or harm them in any way. So even if they are protected, they are still facing like very severe conservation problems. The most important are the illegal, in many cases, target fisheries, because these mobulas are very valuable for their meat. In some places, like here in Baja, they really like to eat machaca, which is the mobula, the meat of the mobula dry and with a lot of salt. So they really like to eat burritos of machaca. And in an international environment, they are very famous for their gill plates. The gill plates are the structure that they use to take out the zooplankton from the water. So you can imagine that these structures are known in the Asian market for their medicinal skills. So that's why they are using it, and they are killing mobulas in many places, like in Sri Lanka, Indonesia, Peru. And then we have another big problem, that is the bycatch fisheries. That means in all the artisanal fisheries and the big industrial fisheries, as the tuna fleet or the longliners, that normally the ones that use gill nets, mobulas are very susceptible to get entangled on these nets and die. And the other problem is the tourist. When it's not regulated or controlled, as you can see here in the picture, this freediver is doing something that, even if it's like a dream for a lot of people, you don't have to do that, okay? Because you are actually really bothering the animal, and you can cause a lot of stress and make that animal never come back to this area. But if we use the ecotourism in a good way, in a sustainable way, it can generate up to $140 million per year, while the gill plate market only generates about $30 million per year. So as you can see, the tourist is more profitable for local communities than the illegal trade of their body parts. So now I'm going to talk to you a little bit of my research, what I did with the mobulas. This was like an introduction, but now let's go to the epicenter. So Baja Peninsula, it's very fortunate because we have actually mobulas on both sides, on the Pacific side and on the Sea of Cortez. But the Sea of Cortez is very famous because it's a World Heritage Site since 2005, and it contains more than 800 species of fishes, and it's between mainland Mexico and Baja Peninsula. So it's a very rich area because it has a very steep bathymetry. That means that on the north part we have, also on the south part of the Gulf, we have very deep canyons that with different currents create a lot of upwelling. So water very rich in nutrients come up to the surface, making the water a little bit green, but also producing a lot of first like phytoplankton, and then we have a lot of zooplankton, and then as you can see on this video, we have thousands of mobulas swimming around. So my research was made in Espiritu Santo Archipelago, which is on the south part of the Gulf of California, in front of the Bay of La Paz. This archipelago is a marine protected area, so there are a lot of restrictions for fisheries and also for the tourists. And since like more than 10 years, ecotourist activity started where you could see mobulas at night, Mobula munchiana at night feeding. And it was very beautiful, but we didn't know anything about these rays, why they were there. So I went to dive there several times, and we start to realize like these animals, even if they are the smallest species of mobula, they are very, very, very small. So we start my project research, and we wanted to know the conservation and the ecological implications of this specific site. For that, we have to study the habitat use, the residency, and the movements of these animals. And during the years of 2006 2017 and 2018. We were doing captures and tagging animals with traditional tags and acoustic telemetry, and also monitoring the zooplankton and the temperature of the water to see the reasons why the mobulas were in this area. So here you can see how we actually capture the animals. It is like we need these fishermen because they are experts. Now they have changed completely and they are just helping in research. But also we use a special net of 2,500 meters. It's made out of cotton, so even if the animal gets entangled on the net, it doesn't hurt them. And every time we have an animal on the net, as you can see, Juan just got in the water and bring the animal. And we have like less than 5 minutes to take all the measurements, to do the traditional tagging, acoustic tagging, and also taking blood samples and genetic samples. So we were doing that very, very fast on the boat and then release the animal back into the water. And they all went happy ever after with their tag. Here you can see that's a baby, it's very, very small. So the different parts of the project, the first one was the traditional tagging. Normally, oceanic mantas and one of the biggest mobulas, you can recognize each individual because they have a unique pattern on their belly. But mobulas, they don't have these marks. So we have to put a tag on each animal to know if we are recapturing the same animals each time. So at the end of the year, we have tagged about 110 different animals. And what we saw is that most of these animals, 65% were juveniles, while 19% were neonates and just 15% were adults. And we even caught pregnant females that we were also able to do ultrasound and see the baby inside. So the size range of the mobulas that we captured was between 49 to 110 centimeters of this wide, so since babies to adults. And also we have recaptures, all from juveniles and neonates, from 1 day to 8 months later. And we recaptured the animals like in the same place that we tagged them for the first time. The other thing that we did is the acoustic telemetry. Acoustic telemetry works in a way that you put an acoustic tag, and the one you can see here. It's very, very small because these animals were neonates and juveniles, so they were pretty small. And we make a small incision on the ventral part of the animal. We place the tag and then close the incision with stitches. And after, we release the animal. So how it works is that the acoustic receiver works as a listening station. So we go diving and we put this receiver on the areas that we think mobulas are going to pass. And then when a mobula that has the tag inside, and if it goes around the receiver like 300 to 500 meters away from the receiver will detect the code of the tag, the hour and the date that animal passed close to the receiver. So each year we go to all the receivers, we take them out of the water, we download the data, and then the fun part is to analyze it. So how we study the residency with the detections. What we do is we divided the days detected of each animal between the days that the receiver was in the water. So that gives us a number between 0 and 1. If it's 0, that means that the animal was never detected, so it's not resident to that area. And if it's 1, that means that it's super resident, like it didn't move from that area. So with Palacios, we had about 60 receivers all along the Gulf of California, but also on the Pacific side of Mexico. and that allows us to track the movements of the animals if they go to any of these places. And if we go a little bit closer to the Bay of La Paz, where we were doing the tagging, you can see also we have around like 20 receivers, and on the red circle is where we have the 98% of the detections of our animals. That means that the small rays, the small devil rays that we have tagged inside the bay, they stay within the bay of up to 145 consecutive days. We also have some detections around the island, but there were like just one day passing, and the main action was happening inside the bay. Here you can see the detections throughout the year, and also here we have the smaller individuals and then bigger ones. And you can see they are very resident to the bay, and these green and blue are outside the bay. So very resident within the bay, and then the red line is the temperature. A very interesting thing is that when the temperature drops, the animals are gone from the area. And in this time of the year, April, May, like we are right now, is the time when we have the adults. So adults come here to mate and reproduce. So probably these babies, the ones that are ready, they go with the adults. And the other thing that we did is zooplankton surveys, because we wanted to correlate the presence of the animals with the temperature and the abundance of the food. So we did a lot of zooplankton tows with this net that we have here for 3 minutes in different places inside the bay to see how is the zooplankton during the day and during the night. And what we find out when we compare the abundance of zooplankton that you have here and the detections here on this other map. So in the shallow receiver, let's say, more inside the bay, we have less zooplankton during the day and also less zooplankton during the night than on the deeper receiver that we have here. But we actually have more detections on the shallower receiver. That means that mobulas prefer to be more protected than to have very abundant food, even if they still have food. But they prefer the protection of the bay than being more exposed to predators. So with all these results, we could establish that this bay is, it can be considered as a nursery area. And that means that for elasmobrand, a nursery area is defined as a place where juveniles and neonates are more abundant than in other places. That these juveniles and neonates stay in the area for weeks or months, and that they use the area across years. Like every year they use the same area. So with these results, we implement a management plan with the authorities in order to organize the ecotourism activities and also the small fisheries activities that were permitted in the marine reserve. So what's the next thing to do? Now I'm working on my PhD, and actually I'm focusing now on the bycatch of the mobulas in artisanal fisheries. So what we are doing is trying to improve the release methods for the mobulas that get caught on the nets of the fishermen. And then with the citizen science expeditions, what we are doing is searching for other mobula nursery areas in this region, because there are probably more bays like this one, where the babies are grown. But the most important thing is that we work with the local fishermen. So they have been helping during the research, and now we want to take them from fishing to guide tourists. So because they are the ones that know everything that happens in the ocean, so they are the best guides to take out. Well, I want to say thank you for all the donors that this project has along the way, especially to Monterey Bay Aquarium that funded the research, and the University of California, Systemar, and Palacios. And any questions, I will be happy to answer.
Awesome. Thank you, Marta. That was awesome. Really, really great. Perfect. So let's see. Does anyone have any questions or anything they'd like to ask? We can type in some questions in the Q&A box if you click the button at the bottom of the screen, and then we can go from there and read some questions out to Marta. Perfect. Let's see. We got a couple coming in already. So Sue asks, I've seen videos and pictures of aggregating rays of different species interacting. For example, monk's devil rays and cownose rays. Do you have any thoughts on why they might do this?
Yes, sometimes cownose rays and Munkiana goes together. It's because they are feeding on the same habitats, we can say. Like they are feeding on different things, but Mobulas, Munkiana like to feed on mysids that is found in very shallow bays, and the cownose rays also feed on small crustaceans that are within these bays. So probably, for my opinion, is that they are going together. The cownose are joining the Mobulas, like protection, because number gives protection to these animals. As you can see on the other picture, orcas normally eat them like un aperitivo. So if they are in large numbers, it's less probably that they will get caught by predators. So I think that's why the two species combine, because they share common habitats and because number gives them a strength.
Awesome. That was really cool. We actually, last year, we got to see that. I'm trying to see if I have the pictures on this computer. If I do, I can share it with everyone to show what Sue is talking about. It's really cool with the cownose when they come in, because they look bright yellow, like almost like gold from the sky when you see it from the drone and everything. It's such a different color to see them, you know, coming in with the Mobulas. Let me see if I can pull it up and just to show everyone what she's talking about. But it's really interesting to wonder, like, why they would do that, like that they can, you know, it's like they're hanging, you know, almost like they're socializing in a way, you know. Let me just put this like this, and then that's the same one. Okay. So here I can share a screen to show you guys what Sue was talking about. So this is something we witnessed last year with the expeditions and everything like that. If you look on the right of the screen, you can see the golden cownose coming with a Mobula. But if you also look inside the school, you'll see the flakes of gold, it looks like, already inside the school and hanging out with them. We actually tracked them from, I don't know, it had to be a few hundred meters away, following this one Mobula in and coming across and then joining into the school, like starting to move in the same spiral that they were moving, and then eventually just becoming part of the school and like hanging out with them, like they just like, you know, like they were joining a festival or something like that. Really interesting. Super, super cool. Cool, thank you for that answer. All right, let me find next question. All right, so we've got a question from Victoria. Victoria says, thank you so much. What are the 3 techniques of monitoring? You said zooplankton surveys, acoustic telemetry, and what was the third one?
Traditional tagging. Maybe I didn't explain. Traditional tagging is when we put like a plastic tag on the animal and it's visible. If you pre-dive with them, you could see the animals that are tagged because the tag stay outside the animal. These tags have the number of the animal and the telephone number from the researchers. If someone for some reason caught the ray, they can call us so we can track their movements and see how they have grown. So that's the traditional tag. And other thing that we did, but I didn't explain here because it's kind of more laboratory stuff, is we did stable isotopes, which is taking blood from the animals and then analyzing the isotopes of carbon and nitrogen, so we could make an idea of where and on what they have been feeding for the last 3 months. So that's another technique. And if you combine all together, that gives you the, it was very fun because at the end, the different techniques gave us the same answer. So we were very sure that it was like a nursery area because the isotopes, the telemetry, and the traditional tagging gave us same results.
That's incredible. That's really interesting. Do they have, like, have you ever tried working with like satellite tags with them or anything like that? Or are they satellite tags too big for them?
With the University of California, they have put mini path tags on Munchiana, but for the juveniles and for the babies, they are way too big. Like satellite tags work on Manta Rays, even in some of the Mobula species, but for Munchiana it's too big. So even if you manage to put a satellite tag, you won't get very good data because probably you will have the data of an animal struggling to swim. So that's why we don't use these super expensive tags.
No, of course, yeah, super expensive. I was just curious if because most satellite tags are so large, and on such a smaller animal, you would think it would be really difficult. So it must make it even more difficult to be able to track them and know that than being able to just pop satellites see where they are going through satellite tagging. Awesome. So Diana asks, do you know how long the young mobulas stay in the nurseries? And do you have evidence of them coming back to the same nurseries as adults to mate?
We have seen that they stay within the nursery for up to 8 months. And we don't have evidence yet because it didn't pass the time for us to see them as an adult. They take them very long time to grow, but the acoustic tags that we have placed on the animals, the battery lasts for 3 years. So if they come back in these years, we are going to be able to detect them. So like that, we could know if they are coming back. But probably it will take more than 3 years for them to reach sexual maturity or to come to this place to give birth.
Amazing. It's like a waiting game, having to wait for them to come back and return and see if they actually do. But it'd be incredible to see if they do go back, to the same places. Awesome. Victoria says, thank you for your response on her previous question. She says, where do the rays lay their eggs? Do they lay it under rocks or around coral reefs or in the mangrove system? Okay, so this probably I don't understand. There are some rays as sharks that they lay eggs, and there are other rays and sharks that they give birth to live animals. So mobulas, they don't lay eggs. They give birth to live and independent and free swimmer animals. So actually it's very interesting in the way that they gestate the babies, because at the beginning the baby is connected to a jaw sac, so like other sharks, and then the baby stays inside the uterine of the mother, and the mother develops like some sort of filaments that nutrients the baby. It's called trophonemata. These structures give a very rich nutrient liquid that feeds the animal. So the baby is inside the mother, actually pumping through their mouth and feeding on this liquid that the mother is giving them. So it's called a placental viviparity. So they don't have like placenta like humans, but they have something like that. So when the baby is ready, the mother just gives birth and the animal is completely free. No eggs. So that's a mystery that we really don't know where they are giving birth.
it's never seen in the wild for any manta or mobula species where actually the babies are born. We think probably around this area because it's a nursery area, probably at depths, but we don't know. We need more technology for that.
Very incredible to find. I mean, you would think if they're not born, say, near the nursery, then they need to travel from birth and know how to get to one of these nursery areas and everything like that would be really interesting to understand, you know, like if they're not born, say, physically in like the bay or something like that, and they're born elsewhere and they need to find their way there, how do they find their way there? And it just brings up, you know, a lot more questions, a lot more things, you know. Super interesting. All right. Jesus asks, would more government support be necessary to support the species?
So here in Mexico, they are protected by a fishing law that restricts their fishing, and they have been added to a new category of protected species, like the whale sharks and the great whites here in Mexico. So it's like the wildlife rule, let's say. So they are super protected. The problem here and many other countries is the enforcement of these laws. So in Mexico, we don't have a lot of observers or a lot of people in the fishermen camps taking account of what the fishermen are bringing. So we've seen a lot of illegal fishing of mobulas within the Sea of Cortez, where they are protected even in marine parks. So it's like the typical problem of a lot of laws on paper, but really not very enforced on the field. So yes, we will need more engagement of the government or more money for the surveillance.
More funding and everything like that. Yeah, I mean, even what we were just talking the other day that there was reports of them being fished illegally just last week from an area within the Sea of Cortez inside another protected area. It's sad to see, and yeah, it makes it hard. It's one of those, the downsides of marine parks is they look great on paper and in politics, but without the enforcement or the funding even to enforce these areas and all that, it's really difficult to you know, make it work and all that stuff to actually, you know, get the protection working that they need. Awesome. So Marina asks, have you ever evaluated the sex ratio of the mobulas inside the bay?
Yes, actually within the bay we had a sex ratio for juveniles and neonates of 1:1. So for one female we have one male. That is a typical feature of the elasmobranch nursery areas, because when they are born with the same probabilities of being a female or a male. But there are other studies with adults, and they have seen that the ratio is always like more female. In some groups are more females, adult females, and in other groups more adult males. But within the nursery area, the ratio is 1:1.
Wow, that's interesting. Really interesting. Enrique says, hello, Marta. Thank you very much for the very nice talk. My question is, how deep do mobulas go in search of food? Do they mostly stay near the surface, or do they go very deep searching for moving plankton?
That depends on the species. For example, Mobula munchiana, the one that I explained you more, they don't go very deep. They stay between the 50 meters, the first 50 meters of water. It's because they feed on euphaseeds and mysids that normally they migrate. The zooplankton migrate during the day and the night. During the night, the zooplanktons come to the surface, and it's normally when the mobulas are feeding. So they don't need to go that deep. Especially these species don't go normally below 50 meters. But there are other species, like the biggest species of Mobula, the Tarapacana, they can go up to almost I think it was 1,700 meters. So they make really, really deep dives. And what they do is they just like glide super fast, and in less than 2 meters, they are from the surface to 1,700 meters. Because during the day is on these layers where the food is concentrated. So they go there and they feed and they come to the surface. And a way that they have adapt to this kind of diving behavior, because when they are going that deep, the water, it's really cold and they need to keep functioning. So what they have is the rete maravillosa, it's called in Spanish, I'm sure in English. And it's a way that keeps their brain warm. Even in very cold conditions outside, the brain is warm, so that allows them to have the sensory functions like the olfactory, the eyes, like very functional. And this red maravillosa, it's like a contracurrent exchange of blood with a lot of oxygen and blood with low oxygen. So blood more cold and warm, and it's exchanging the heat, and that warms up the brain. So yeah, it depends on the species, but it goes from 50 meters to 1,700.
That's incredible. I can't imagine it diving 1,700 meters. That's mind blowing. And for those that are joining from the US, anything that's like what, 3,000, almost like 5,000 feet, something like that underwater. That's unreal. And to do it so fast, like to be able to just quickly dive down, feed, and spend time down there, and then come back up is just wow. It's got to put them as like, up there with some of the deepest diving animals out there. It's incredible. Awesome. So Sue asked, do we know where the mobulas go when the temperature drops? Have your tagged rays been detected anywhere else outside of the Sea of Cortez?
For the tags that I have tagged, like my babies, they didn't go out of the Sea of Cortez, but they have been a little bit farther out to Cabo Pulmo, which is another marine protected area, like not sure how many kilometers, but like 3 hours away from where we are. But probably the adults are making longer moves and in shorter time. For Mobula munkiana, I would say that normally they stay within the Gulf. Like the populations are moving from the south to the north of the Gulf. For other species, as the Mobula mobular and thurstonite, they tend to go to the Pacific side, depending on the time of the year, because they are following the zooplankton. So since they are bigger, it's easier for them to travel longer distances. It's like the oceanic manta rays that they can move vast distances in a very short time.
Mm-hmm. And do the munkiana, do they follow like a specific temperature range? Like, do they try to stay in like a specific temperature at all times?
So what we've seen in the nursery area is that they prefer a temperature between 25 to 29 degrees. It's when the babies show more residence inside the baby. But of course, then we have here the aggregations of adults, and sometimes the water is like 19 degrees, 20 degrees, so they can stand like lower temperatures.
No, it's incredible. And as I was telling you the other day, like when one of the times we had when we were out with them, where we seen following a school, and it felt like they were like almost following the warm water, because you'd feel like a colder water current come in, and they would like move away from it and like turn around and go back to warmer water and then come back. I was like, oh, they're just like me. They just want to stay warm. But then we've other seen too is, I mean, we've had last year where we had the vortex feeding and that stuff, where we had a giant school, and you know the water outside of the school was, you know, say 24, 25 Celsius, something like that. But then inside the school, this massive tower of a school, where it was, you know, there was like a 3-degree temperature range. It was down into like 21, 22, where it felt almost like they were bringing the colder water up within them and all that. It was really interesting. Awesome. So Jesus says, congratulations on the extraordinary presentation. What do you think of the collaboration with local fishermen for the preservation of species?
Well, I think it's essential. Like, we can do whatever we want, but if we don't get the involvement of the local communities or the local fishermen, it won't do anything, just papers. So it's very important. Sometimes it's hard to get their attention in the first place, and more when you are a woman and when you are from a foreign country. But if you get to engage with them with other things, then they are really essential for any research that you want to do with live animals in the sea, because they are the ones that know where the animals are, how to catch them, where we can find them. So I think it's essential to work with local fishermen in research and also in ecotourism.
No, I completely agree with you there. I think it's the way forward. You know, I mean, without the support of the local communities, the projects are just, like you said, it's just paper. You know, I mean, there's no real support or anything like that. And at least involving them, one, it helps to show, you know, how things can be made more sustainable within that area instead of taking them out of the water, keeping them in the water, but also getting them behind protecting the area and wanting to, you know, work with you, like work together to better protect their home and all that. Cool. So Christina says, thank you for this very interesting talk. What was your favorite experience while working with these creatures?
Well, I have like different experiences. With mobulas, I really enjoy like free diving with the munkianas, with a huge school of munkianas, because you are at the surface, and when you are at the surface, they are chilling around you, so you can observe them. But when you go down, it's like they all fly around you, and it's just amazing. So every time that I find a huge school, I just jump in the water, because that feeling of being surrounded by flying fish, it's amazing. And with the manta rays, last year I had the opportunity to go to Revillagigedo, which is a very famous dive area where the mantas are extremely curious about divers. So we could dive for like 50 minutes with an animal just following you, surrounding you, going up to you. So it's amazing the interactions that you can have with these animals. You could see that they are very curious, and that means that they have intelligence.
Awesome and beautiful. Jose says, is there a predicted effect on the localization / survival of mobula populations derived to the rise in sea temperatures from climate change?
More, well, I think it really will affect the prey abundance and the prey availability. So probably that will affect the mobula more than the mobula survives. Like, it will affect where we can find them. They will move, that's for sure. If the food, they are just searching for the food, so they move between habitats until they find a patch of zooplankton. So if the water starts to warm up, the zooplankton is going to be in colder water, so probably they will go a little bit more north and north and north each time, but we don't know.
And Sue asks, do you know if the mobula populations in the Sea of Cortez are stable, or are their numbers decreasing or increasing?
That's a big problem that we have all the time. Is there's no studies on the population's status. First, because it's very difficult to count them when they are in thousands, and the other is that for the other species, they are very elusive. Like, very difficult to see. But what we know is in 2000, well, no, in 1980, mobulas and mantas were super abundant on the Sea of Cortez. You can hear a lot of talks of Sylvia Earle saying that this was paradise for the mantas. You could dive anywhere and you could see many, many mantas and thousands of mobulas. And then in the 90s, the fisheries of the mobulas and the mantas started to go up. And since then, until almost 2 years ago, you couldn't see a single manta ray, oceanic manta ray, in the Sea of Cortez. It was super, super rare to find them. With the mobulas, I think you can see on the Mobula munkiana, for example, that the groups, as the fishermen said, you know, like the groups are getting bigger, but it's very difficult to tell. But for the oceanic mantas, the population collapsed at some point at the end of the 90s, and it's just until 2 years ago that they came back. And this year they didn't came back at all. Well, not as many as the year before. So I don't think the populations are recovering very much.
No, it isn't true. That's the reason why the IUCN upgrade, or well, I don't know if you could say upgraded, but like why Mobula munkiana was moved from near threatened to vulnerable is because of that, right? Because of the data deficiency in the populations.
Mm-hmm.
So, I mean, sad to say. I mean, and it's crazy because we get to see them in these gigantic schools and everything like that. So it's hard to, you know, like connect that, you know, yes, we see them maybe in schools of 10,000 or something like that. But the reality is in total is that the population is most likely still decreasing due to, you know, bycatch and fisheries and these kind of things and all that. So it's really, it's sad, you know what I mean? Because it's such an incredible experience in something that there's, you know, nothing like it on anywhere on earth, you know, that's just is mind-blowing. And to know that, you know, in a few generations it may not exist anymore is just scary, you know.
Mm-hmm.
So, awesome. I think that's all the questions we've got from everybody. So thank you so much, Marta. Thanks for joining us today and everything like that. Right now, we should be out on the boat together working with the Mobulas, but we're kind of both stuck at home with the quarantine and everything like that. Hopefully soon we get the okay to get out on the water and go run some citizen science expeditions this season. So fingers crossed for that. Hope to see you very soon. Thank you everybody for tuning in today. Tomorrow we have the day off, and then we're back on Thursday with Martin from Coral Guardian. Friday we're back with the Jack from Project Aware. Then also on Saturday we have something special coming up too. We've got a Project Aware whale shark conservation course that we'll be teaching on Saturday morning. Actually, I will be teaching it with Ellen. So if you want to learn something about whale sharks and how we work to protect them and all that, make sure you check that out. You can get more info on the Dive Ninjas website. Otherwise, we'll see you guys all back on Thursday. Marta, thank you so much again.
Thank you for inviting me. It was very fun.
Thank you. It's been awesome. All right, everybody, have a wonderful day, and we'll see you on Thursday. Take care. Have a wonderful day.
Bye.
Bye.